Good evening and good morning. Thank you all to join our third webinar. So my name is Qing. I am one of the members of the Mekong One Health Innovation Program management team at Michigan State Universities. Along with us today are the other two team members, Dr. Furquan Irfan and Dr. Ramji Gamir. And also on our panelist, we're lucky to have Deputy Assistant Secretary Jim Levy from the U.S. Department of States. He's going to give remarks later. And we have Victoria Harrison, who has been helping us and put this program together from the State Departments. And of course, our speaker, Professor Shannon Manning, will be our key speaker today. And we have our new program manager from the State Department to monitor, supervise and work with our team is Mr. Wayne McKenna. And also that's our Dr. Phuc from Vietnam, who is our country advisor, and he is a director of the Vietnam One Health University Network. He's a great help from the beginning. Thank you so much. So before we start today's program, I have a couple announcements just to make sure people understand what is going on. One of our survey post-webinar survey from the past two webinars is some people wants to be able to speak because under the webinar mode, audience attendees are muted automatically, but you do want to speak, please raise your hand or put your request in the Q&A, and we will put you on mute and you can talk. The other thing is, as usual, we sent out post-webinar survey. We hope everybody can complete that survey, give us feedback so we can improve and make things, future events better. And the other one is we're going to do another round of introduction of our program because we have new audiences. That's why for the older audiences, please bear with us. And the last one, but not least, is we have a new email address. In the past, I sent out the invitations from my personal email at MSU, but our program has an official mohip@msu.edu. From now on, the invitations will send from mohip. Please understand this is our official email, so don't delete the emails if you see that unfamiliar email address. The last one today is please do post your comments and questions on Q&A. Dr. Furquan Irfan will monitor the questions and we can discuss or address those questions at the end of today's webinar. So without further ado, I will give this floor to my colleague, Dr. Gamir, to introduce DES Levy and our presenters today. Dr. Gamir.
Can you see my slide, Ting?
Yes.
All right, got it. Thank you so much, Ting. Thank you, Furquan, and thank you everyone for attending this program. This is the third webinar in the series that Mekong One Health Innovation Program has organized, and we'll be organizing monthly webinar every month in the future as well. And first of all, I would like to thank everyone joining this webinar, especially our guest today who will be giving remarks shortly, Deputy Assistant Director, Department of State, Jim Levy, and also our speaker today, Shannon Manning, for your valuable time being available today. I'll quickly walk through what this Mekong One Health Innovation Program is. It will be very short, I won't take much time. And as Ching mentioned, many of you have already gone through it. So those who have already seen it, it will be a repetition. Bear with me for a few seconds. There are three objectives of this program. This is U.S. Department of State Bureau of East Asian and Pacific Affairs funded cooperative program. And the first objective is create a transnational network between Mekong and U.S. researchers working on health security, especially focusing on one health area. The second one is identify the challenges and gaps in Mekong region, especially health security issues and One Health, and develop and offer a course on health security, One Health, and zoonosis. Third one is provide funding to Mekong researchers to carry out research pertinent to the health security issues facing Mekong countries. And for the first one objective, we are organizing monthly webinar. This is one of those webinars. And for the second one, we will soon be hosting or organizing a short course for targeting our Mekong researcher. The third one is we'll be providing the funding to the Mekong researcher after we soliciting the call from the researcher in there. So these are the two webinars we already did, and third one is the webinar today we'll be doing. So this is the overall structure of the Mekong program that we have. And as Ching mentioned, we have very fortunate enough to have the country partners and collaborators here, and who represent International Advisory Board. Dr. Fu, for example, he is the International Advisory Board member. And country coordinator, we have two country coordinators from Thailand. One is from Laos and Vietnam. And they are kind of helping us implement this program in the field. And we also have US health experts whom we have been constantly communicating and seeking their help to leverage their expertise in this program. And also, importantly, Mekong researcher and stakeholder are the skeleton are the primary beneficiary of the program, whom we have been trying to reach out, and we will be trying to reach out to more of them as we keep implementing this program. This is the short info. If you have any questions about the program, this Mekong One Health Innovation Program, we'd be delighted to answer your questions. And please email us or call us or contact our country coordinator. This said, I would like to now briefly introduce our today's guest before he delivers his remark. And we have with us Jim Levy. He serves as the Deputy Assistant Secretary in the Bureau of Ocean and International Environmental and Scientific Affairs at the U.S. Department of State. Jim Levy is acting Deputy Assistant Secretary in the Bureau of Ocean and International Environmental and Scientific Affairs at the U.S. Department of State. He has served several assignments in China, most recently as American Consul General in Guangzhou from 2018 to 2021. His overseas assignment also includes Beijing, Taipei, Berlin, Mumbai, and Islamabad. In Washington, Mr. Levy has served assignment at the Bureau of East Asian and Pacific Affairs, Global Talent Management, Consular Affairs within the Department of State, as well as on detail to the Capitol Hill office of a member of the U.S. House of Representatives. A native of the state of Louisiana, Mr. Levy studied at Yale University. He is married to Bonita Shun, a chamber music performer and management analyst who is originally from Vancouver, Canada. Mr. Levy and Mrs. Shun have one daughter and two sons. With this brief introduction, and this is our privilege to have him in here. Mr. Levy, please go ahead, and we are excited to listen to your remark. Let me unshare my slide.
Thank you so much, Professor, for your kind introduction. It's a privilege for me to be here. Thank you for your invitation to join the webinar and to meet with this outstanding group of researchers. I've been an American diplomat for several decades, as you just heard, and much of my work has focused on American relations with countries in East Asia. I've been privileged in particular to work with counterparts, academics, and others in China, Taiwan, and representatives of Vietnam, Malaysia, Singapore, many other countries in the region. In my current capacity, as mentioned, I'm a Deputy Assistant Secretary of State overseeing the Office of International Health and Biodefense here at the State Department. And in that capacity, I work with nearly 30 people from a wide array of backgrounds. Some are government experts, some are laboratory scientists, some are epidemiologists. And some of them are on this call with us today. They have been instrumental in putting together this MOHIP program, together with others from our organization here at the State Department, plus your excellent team at Michigan State University. You're in good hands. At IHB, my office, we address the challenges wrought not only by acute headline problems such as COVID-19 and polio eradication, but also those slower moving yet still deadly challenges that erode health security and that threaten all of our personal, community, and societal well-being. Antimicrobial resistance, or AMR for short, is one of those problems. The statistics are staggering. Already, right here and right now, we're seeing 1 million deaths per year that can be attributed to AMR. That number could grow to as much as 10 million per year as AMR intensifies and our ability to detect it improves, perhaps by 2050. By that time, the economic costs could reach 3.8% of global GDP. The stakes here are mind-boggling. Nearly all of us have been to a hospital or a clinic in the last few years, either as a patient or as a caregiver. Virtually all procedures in those settings, from childbirth to outpatient surgeries to major operations, require antibiotics to maintain safety for workers, patients, and caregivers alike. Losing access to those tools would completely subvert the practice of medicine as we know it and would make even routine procedures deadly. In short, that is a matter of global security and prosperity for all of our countries, ripe for diplomatic engagement and for international cooperative programs like MOHIP. Now, fusing up health and diplomacy is all to the good, but AMR is extra tricky because it requires stakeholders beyond those fields. As we will hear tonight, there are important aspects of the AMR challenge that arise from not only the human health sector, but also from environmental management, food production, and the broader economy. AMR, much like zoonotic disease and climate-sensitive disease issues covered elsewhere in this webinar series, requires a One Health approach, bringing together human health, animal health, and environmental health sectors to solve problems collaboratively. One Health is central to American foreign policy. It is part of our approach to health security. The concept is enshrined in many of our key directives and statements from President Joe Biden, Secretary of State Tony Blinken, and many other officials. While there are many ways to implement One Health, events like this, hearing from experts, answering questions, exchanging information, and meeting with others from different backgrounds in different countries and different career paths is vital. So I am delighted to be here today, and I'm thankful to the MOHIP team for the invitation, and I'm looking forward to hearing Professor Shannon Manning's remarks on this fascinating and frankly hair-raising topic. Thank you so much for inviting me.
Thank you so much, Jim, and for your excellent remarks. And we look forward for your advice as we keep implementing these projects. And with this, let me move ahead and, oh, sorry. Let me introduce today's speaker, Professor Shannon Manning. Shannon Manning is a Michigan State University foundation professor in the Department of Microbiology and Molecular Genetics. She earned her MPS and PhD from University of Michigan in 1998 and 2001, respectively. For her postdoctoral training, she served as an emerging infectious diseases research fellow through the Center for Disease Control and Prevention and Health position at University of Michigan and Michigan State University. She joined the current institutions as a faculty in 2010. Her research focuses on molecular epidemiology, evolution, and pathogenesis of bacterial pathogens, including diarrhea E. coli and Streptococcus agalactiae. She has also been determining how pathogen and antibiotic impact the gut microbiome and contribute to antibiotic resistance. She has contributed to over 100 publications, served as an editor for scientific reports and microbiology Spectrum Journal, and was recently selected as an American Society for Microbiology Distinguished Lecturer. Since joining the faculty in 2010, she has mentored 25 undergraduate students, 21 graduate students, and 13 postdoctoral researchers. With this, we are delighted to have Dr. Manning here, and floor is yours, Dr. Manning. Please go ahead with your presentation.
Okay, can everybody see that okay?
Yes, we can, Dr. Manning.
Okay, great. And thank you so much for the kind words and for the invitation to speak. It's really a great honor, and it was a wonderful overview that was just given. So that's a great segue into my talk here for you today. So I'm really going to focus on antimicrobial resistance in general, and then I'm going to move in to talk a little bit more about foodborne pathogens and some of the work that we've been doing in my lab at Michigan State. And so, as was stated, we know that antimicrobial resistance and antibiotic resistance is really a global health concern. And so antibiotics are really critical for us. And as was eloquently stated previously, these have been around for many, many years, since the 1940s, and they've really been important and critical for treating infections in not only humans, but also animals. And so when these drugs no longer work, it becomes detrimental to us. Because we need to continue to find more drugs, and it puts us in a bad situation. And so more use of antibiotics will just contribute to more resistance. And so hopefully I can tell you a little bit why that happens from an evolutionary standpoint. And we do know from many different studies that resistant infections are very difficult to treat. This is because patients, for example, will come to the hospital and they'll be given one drug. And if they have resistance, then the drug won't work. And so the patient just gets sicker and sicker. And so a lot of times they have longer infections and longer, lengthier hospital stays with greater medical costs. And as was stated, these resistant infections are actually a leading cause of death now. And some numbers for you, just in 2019 alone, it was estimated by the CDC that over 2 million infections were caused by resistant either bacteria or fungi, contributing to over 35,000 deaths. And so unfortunately, the number of resistant infections keeps increasing. And I thought this graph was telling. This was actually just published last year, but it was based on data from 2019, where they identified that there were 6 primary pathogens that were really responsible for about 80% of the 1.27 million deaths linked to resistant infections. And so when you scroll through this list, you can see E. coli here is at the top. And this is one of the pathogens that I study in my lab. And you can see a significantly greater number of these infections were actually resistant, as indicated with the orange bars. And that's relative to those that are susceptible to drugs. And so this is a huge problem for E. coli. But what we're not sure of which types of E. coli, that when you think about E. coli as a whole, there are so many different varieties with different virulence factors that are linked to different infections. And so it's important really to understand this pathogen population entirely. What is more worrisome is more recently, just due to the COVID-19 pandemic, we've actually seen about a 15% increase in these resistant infections. And a lot of that is due to a misjudging, and inappropriately treating patients who might have the viral infection with antibiotics, leading to increases in resistant and selection of resistant bacteria in the gut. And so we know that this selection, or this antibiotic use, is really the key factor that is important for the development of resistance. And so just to highlight how this happens, it happens through natural selection. And so what happens is you have this bacterial population here. And so you can see that the majority of these bacterial cells are orange, and they're susceptible to a given antibiotic. But you might have a few with either innate resistance or maybe a point mutation that allows them to resist a certain drug that's given. So when you add that antibiotic to this population, you're basically killing off all of those that can't survive, and you're selecting for the population that can survive in the presence or that resists this drug. And so eventually those are going to proliferate and multiply, and then you end up with this population of mainly resistant bacteria. And so this is a problem because we know globally that antibiotic use is actually increasing, and more so in specific parts of the world. And it's not only increasing in human populations, but also in agriculture and other industries as well. And so that really highlights the importance of, looking at this topic with a One Health perspective. We know that antibiotics are used in these different industries and environments as well. And so that's actually highlighted here. We know that antibiotic use is widespread. Antibiotics are often given to livestock or food animals, animals just for treatment, but in some cases for prevention of specific infections. And those antibiotic residues can get into food products, and all at the same time, it can be selecting for those resistant bacterial populations. And so those are the ones, even when these residues are present, those are the ones that are going to proliferate. And so it can be excreted in the feces. It can get into wildlife. Plants can incorporate these antibiotic resistant bacterial populations up through the roots and into the leaves. And of course, those can be ingested by individuals. We also see a great deal of use in the hospital system, where individuals are getting these antibiotics. Those are also selecting for resistant bacterial populations. These individuals might go home, continue taking antibiotics, or maybe their children are taking antibiotics from an outpatient setting. And then those bacterial populations can get transferred between them, also through the sanitary system and the sewage system. And then that can get into the waterways, where you see wildlife picking up these different antibiotic resistant populations. But then at the same time, we see treatment and antibiotic use in domestic animals as well as aquaculture. And so all of these things kind of combined are really putting this huge selective pressure on these bacterial populations to evolve and survive in the presence of these drugs. And so we know that this is really critical for the emergence of new antibiotic resistant bacterial pathogens. And so evolution is the key process. And the mechanism for the emergence of these new resistant bacteria is through horizontal gene transfer. And so that's just really the transfer of antibiotic resistance genes from one bacterium to another. And this is a very rapid process. It can really just take one cell that might be susceptible to rapidly transform into a resistant cell just by acquiring a gene that allows it to resist the antibiotic given. And so the way this occurs is through three mechanisms, through transformation, when you have death of a cell, with the red here indicates an antibiotic resistance gene. And so when the cell dies, you get DNA particles and fragments released that get taken up by recipient cells and incorporated into their genome. And so then this renders this cell resistant to antibiotics. This can occur through transduction, or these little phages. Bacteriophage can acquire the resistance genes and insert them into a recipient cell for the same effect. Or it can occur through conjugation. And a lot of these really important, clinically important antibiotic resistance genes are found on plasmids. And these plasmids can get transferred between cells. And so when new cells will acquire a plasmid horizontally, then this cell population will become resistant, as well as the all of the offspring. In addition, we know that some of the antibiotic resistance that we see can occur due to having point mutations in certain genes. And so not really transfer of genes across bacterial cells, but an individual mutation that can render a cell resistant. And those are a little more scary because you don't necessarily need the antibiotic use to select for those bacterial populations. Those are going to proliferate because they're going to be, that mutation is going to be found in all of the offspring. And so really, to understand kind of the magnitude of the problem, we really rely on our surveillance activities. And this helps us figure out, where the key problems are, which populations might be at higher risk than others. And so surveillance activities are really important. And we do this a lot for the foodborne pathogens. And so that's what we're doing in my lab. But it's very similar to what we do in the US through the CDC, where they're monitoring for foodborne infections through the FoodNet system. And this involves 10 different sites in the US, and these are highlighted in this dark green color. And so when these infections are identified, any type of foodborne infection gets identified in these sites, they estimate what the incidence and prevalence of these infections might be for the entire US, just based on the geographic location. And so it's kind of a representative sample. But in Michigan, we are not part of FoodNet. And so before I came to MSU, it was not necessarily clear how many infections we had and whether or not they might be resistant to different antibiotics. And so that was kind of what we sought to do. In addition to FoodNet, the CDC, in collaboration with the FDA and the USDA, has also developed the NARM system. And that basically takes all of the isolates, the bacterial isolates that come from this FoodNet system, and it allows the lab staff to actually characterize these isolates to determine how many are resistant to commonly used antibiotics. And so this gives us an idea of kind of a baseline estimate, depending on the time, of how many infections are resistant at a given time. And then you can evaluate changes in that frequency over a period of time. And so that's what we're doing in Michigan. We started collecting isolates in 2001. And so we've been able to evaluate over changes over a 20-year period. We've been examining Campylobacter jejuni, which is a leading cause of foodborne infections, as well as Shigatoxin producing E. coli and Salmonella. And so I'm going to call Shigatoxin producing E. coli S-Tech just for simplicity. But this is just a variety. It's actually a very potent variety of E. coli that we see. And so this typically is a foodborne infection, but it didn't emerge until the 1980s. And so our goal was really to determine how frequently it was found in Michigan, which is the state where it emerged, and whether or not it was resistant to any of the known antibiotics that we can use. And so Other goals were to identify risk factors for infection, as well as to better understand how these pathogens impact the gut microbiome. And so we know that individuals who get these foodborne infections get very sick. They have diarrhea, and we know that it takes them a very long time to recover. And so how are their microbial communities in their gut changing when they have these infections? We also wanted to conduct comparative studies by collecting isolates from different food animals for comparison. And so we're using some genomic tools that I'll tell you about in a minute. So to focus a little bit more on STEC, I thought I would start at the beginning and how it emerged. And the interesting thing about this is STEC actually emerged by horizontal gene transfer, similar to antibiotic resistance. And so there are a lot of parallels when you think about increases in virulence of a pathogen, as well as increased ability to resist antibiotics. So in 1982 in Michigan and in Oregon, there was an unusual illness that was reported in about 47 individuals. And these individuals were presenting with grossly bloody diarrhea, and then they had severe, extremely severe cramping. And what they found when they did a case control epidemiological study was that the cases were more likely to have eaten hamburgers at a specific restaurant. And so because of that, they were able to extract the bacteria from one hamburger. They had one frozen hamburger patty left, and then they had the stool samples from several cases. And they were able to uncover a new pathogen at the time. And it was E. coli 0157H7. And so now this is obviously a very common pathogen. It's ubiquitous. We see it often causing large-scale outbreaks in the US and elsewhere. And so once it emerged, it was here to stay. But again, it emerged through horizontal gene transfer by transduction. And so it was infected by a bacteriophage that carried genes that allowed it to produce a very potent toxin. And this toxin is called the Shiga toxin. And so this is also a rapid evolutionary process where you can have one bacterial cell acquire the genes that allow it to produce this toxin. And then that population gets selected for and proliferates. And then all of a sudden you have a whole population and maybe even different types of E. coli that can possess these genes and produce the Shiga toxin. And so we know that the Shiga toxin, when it's produced in the intestinal epithelium, this just represents an epithelial cell. It can attach and then produce this toxin that can get into the bloodstream and travel to the kidneys. And it can cause hemolytic uremic syndrome, which can cause kidney failure and death, particularly in young children. And so it's a very potent bacterium and a potent pathogen. And so what we've seen, as I mentioned, is that this diversity of this whole pathogen population has actually increased over time because different types of E. coli are acquiring these Shiga toxin genes. And so by acquiring these genes, it basically takes it could be a commensal E. coli and immediately turns it into a pathogen. And so what we did is we examined over 800 isolates from patients with these STEC infections. And we looked at the types of the serotypes, or the 0157 or other O types that these pathogens actually possessed. And this is a little bit busy, but hopefully it allows you to understand how diverse this pathogen population actually is. And so back in 2001, these different colors just highlight the number of different, or the percentage of different O types, like 0157, 01, 091, for example, which are all represented by different colors. And so what's the most important thing to understand is that if you follow this line, you can see that this represents the total number of serogroups that were identified in a given year. And so you can follow it from, 5, and that's on this axis here, from 5 all the way up to almost 30 different serogroups that we see that have been linked to infection. And so the diversity of the pathogen population itself is actually increasing significantly due to this rapid transfer of these Shiga toxin genes. And so we also wanted to see whether the same was happening in the cattle reservoir. And so a bunch of students, and this is my lab manager here, went out and sampled over 1,000 cattle from 12 different herds. And we recovered over 522 different bacterial isolates from 175 animals. And what we found was that 0157, which emerged in the 80s and really caused the majority of illnesses that we had seen in the US, was really kind of a rare population in this whole group of isolates. And so the majority of these were diverse, representing different serogroups. And what we found is that there was readily transmission of STEC within animals sharing a herd, a pen, and that many of these animals were persistently shedding E. coli, this STEC, into the environment. And so you can see here, this highlights the different serogroups of the bacterial isolates that we recovered, only from 26 cows, followed over four time periods. And so what was interesting is we uncovered a lot of those isolates that we typically see in human infections, which is worrisome. But we also found that there were some that had unique traits that made them better able to persist in these cattle. In particular, this group here, the O6s were significantly more likely to form these strong biofilms. And so that we think is important for survival in this cattle reservoir as well as the environment. And so when they're persisting, they're also much more likely to acquire genes from other bacterial species. And so this would be the same concept for antibiotic resistance gene acquisition in these environments. We also find evidence of transmission of these E. coli populations between cattle and deer, kind of highlighting the importance of One Health and highlights the problem essentially. And so our next question was to see whether any of these patient isolates were actually resistant to antibiotics that are commonly used to treat E. coli infections. And interestingly, we know that antibiotics are not actually recommended for patients with STEC infections. And the reason is because antibiotics induce Shiga toxin production. And so they could potentially make the patient sicker. But even though they're not resistant, we wanted to see how many of these pathogens, because they were E. coli, were actually bringing these resistance genes into the human gut through these infections. And so in order to do that in the clinical lab, we utilized different techniques. The first was disc diffusion, where you take the bacterial isolate, you plate it on some selective agar, and then you put these little discs on here embedded with the antibiotic. And so in this case, you can see multiple antibiotics were tested. And so when you look at the bacterial growth the next day, what's important is to identify those drugs where bacteria can grow right up to the edge of the surface. And that would really indicate that this bacterium, whatever isolate this might be, is fully resistant to this antibiotic, whereas this one would be fully susceptible. And so we know based on the size of this zone of clearing, whether or not these would be classified for each drug as resistant or susceptible. You can also determine the actual concentration or the minimum inhibitory concentration that completely inhibits bacterial growth. And so you can see here, this represents an E-test strip. and it is a gradient of the antibiotic concentration. So here at the top, you can see that you have a very wide range of clearing between the bacterial growth and the antibiotic, which is getting out into the media. And so this is the highest concentration. And so you really look for this point where or this intersecting line where you see growth of the bacteria. And so that would be classified as the minimum inhibitory concentration for this drug. And so when we screened, over 800 isolates from 2001 to 2014 in Michigan, we found kind of an interesting but surprising trend. So even though antibiotics are not used to treat these infections, a lot of these infections a lot of these bacteria were actually resistant. And so they were resistant to more than one drug. And there was a significant increase over time to ampicillin for ampicillin resistance and trimethoprim sulfa. And so there are some fluctuations that we saw, which are kind of interesting over time. But if you do a test for trend, then the result is actually a significant increase. And so we're at the point now where we're going to try to continue to see where we are from here. What was also interesting is that the majority of the resistance is in the non-0157 isolate. So all of the other serogroups besides 0157. And if you recall, those are the ones that we found were more abundant in the cattle population that we were sampling. And so it suggests that it's possible that a lot of these are resistant due to survival or pressures in the environmental hosts. When we compared the frequency of our resistant isolates to those that were recovered from NARMs, you can see here there were differences. So this is the percent resistance. This is to ampicillin. So you can see in Michigan, we had a significantly greater frequency of ampicillin resistant STEC relative to the whole nation, and all those other sites that I showed you that were getting sampled in NARMs. And so this is actually really important because it really highlights the importance of different geographic locations, in that every environment and every location might have unique selective pressures that are actually selecting for different resistant populations. And so when certain and different resistant populations get selected for, you're going to see varying frequencies of horizontal gene transfer of those resistance genes into different bacterial hosts. And so what we also wanted to do is to classify the antibiotic resistance genes using more advanced molecular methods. And so now the CDC is recommending for all of these foodborne pathogens to subject them to whole genome sequencing. And so the protocol is highlighted on the website, but essentially you get a sequence and you can identify the presence of different resistance genes within a genome. And you can also identify whether there's variation or sequence or genetic variation within those resistance genes, which could be important as well. So among those non-0157 genomes that we examined 208 of them. We identified 33 completely different horizontally acquired resistance genes. And so if you remember in the prior graph that I showed you, it showed that we had a very low level of antibiotic resistance. So it was only about 4% to ampicillin. But we didn't test for any of these other drug classes. We didn't test the quinolones or the macrolides, but we did test the beta-lactam, the ampicillin. And interestingly, it wasn't just one resistance gene that all of these isolates had. You can see there were multiple beta-lactamase genes. And these are really important for other reasons that I'll discuss now. So even though antibiotics are not used clinically to treat these infections, we know that these pathogens are actually bringing these genes into the gut microbiome. And some of the genes that we were detecting are these beta-lactamase genes. And so this just highlights the mechanism of beta-lactamase action. And so penicillin, for example, and ampicillin and all of the beta-lactam antibiotics have these beta-lactam rings. And the beta-lactamases are just enzymes. And so these genes encode for these enzymes that actually attack this ring, this beta-lactam ring, rendering the drug completely ineffective at killing the bacterium. And so circulation of these genes that encode these enzymes is very worrisome, especially because the beta-lactam drugs are, there are so many. And so you can get extended spectrum beta-lactamase production that can actually target and render a bunch of different antibiotics resistant, or a bunch of resistance to different antibiotics. Because of their importance. And so these have been targeted by the CDC as a really important group of, Enterobacteriales. And so any of these bacterial populations, E. coli included, that are possessing the genes for these enzymes are very worrisome, because these enzymes and these genes can get transferred so readily into different bacterial hosts. And so this is actually what we found. We were able to show that this is happening. We found when people are sick with foodborne infections, they have this expansion of Enterobacteriales in their gut. And so this population that is important for beta-lactamase production. And so you can see here, this is in 200 patients with enteric infections. We did some 16S sequencing to classify the gut microbiome. And we looked at the abundance of different phyla in the gut. And so in patients, we found this expansion of this proteobacteria population, which is a member of Enterobacteriales. And so when you're sick, you all of a sudden get this expansion of this population, in this bloom of this population in your gut, that is more readily able to acquire resistance genes. And so when we looked at the resistance gene population by deeper sequencing methods, we did actually find a greater abundance of beta-lactamase genes. And we also found that those beta-lactamase genes persisted even after the patients recovered. So we followed that same group of patients, and even 8 to 6 months later, we got a follow-up stool sample, examined the DNA composition of the microbes, and we found a greater abundance of these beta-lactamase genes within the gut microbiome, suggesting that they're kind of there. And so when you get this expansion of this great bacterial host, you're going to get a lot more horizontal gene transfer in the gut, potentially creating opportunities for the emergence of new opportunistic pathogens. We did a very similar study in the gut microbiome of dairy cattle given antibiotics, and we found the same result. These beta-lactamase genes were persisting even after antibiotics were taken away, suggesting that these genes are getting transferred readily in the presence of antibiotics, and so they're getting selected for in this bacterial populations. So just to summarize, hopefully it's very clear that antibiotics resistance is really important. It's a global health concern. We know that these resistant foodborne pathogens have emerged because these resistance genes are so readily transferred between different bacterial cells and species, and they're getting selected for by the use of antibiotics. And we know that surveillance is really critical to understand the problem, to figure out whether there are new pathogens emerging because they've acquired new traits like the shigatoxin genes, or they've acquired different phenotypic characteristics like antibiotic resistance to clinically important drugs that are commonly used in human medicine or veterinary medicine. And so in Michigan, we know that I showed you data that the STEC population has actually diversified and these antibiotic resistance frequencies are increasing. And so it's really important to understand what's going on in other geographic locations and other populations as well. And finally, we saw that there was this persistence of some of these really important clinically important resistance genes in the gut microbiome, even after infection. And this, of course, can result in the emergence of new resistant opportunistic pathogens that we're seeing regularly. Some of these can cause infections when you least expect it. So for example, in a hospital setting, if your gut microbiome harbors these resistant populations and you see transfer into others and you become immune compromised, then you might get another infection or an opportunistic infection. And so the ease of transfer makes it very concerning. And so really combating this problem requires this really integrated One Health approach with people talking and collaborating as you are all doing today and through this series. And so I think it's wonderful for everyone to share their ideas and perspectives and that people are really listening. And so some possible solutions that the CDC has highlighted is enhancing surveillance, which is what we're trying to do, at least in Michigan. Improving education, maybe making people understand compliance and the importance of not taking antibiotics if you're not sick with a bacterial infection. So judicious use that can actually prevent that selective pressure. And also limiting use of specific antibiotics that are really kind of the last resort drugs that are used in human health, where we have no other drugs available. So limiting use to just those types of infections will be important for the future. And finally, just a shout out to some of the students and former lab members who contributed to some of the work that I showed you. I've highlighted some pictures here with those indicated in bold, and I tried to present them throughout, as well as my collaborators at the health department in Michigan, as well as the vet school at MSU, and of course my funding. And so thank you very much for your attention, and again, the invitation. It's great to be here.
Hi, Furkan, go ahead.
Thank you, Dr. Manning, for the talk. We will now open the floor for Q&A. I saw there was a question from Kosal, from Cambodia. Kosal, feel free to unmute and ask the question yourself.
Thank you so much, Professor, for having me. And thank you to the speaker. I have two questions. Because refer to the animal because they have. So can you get me?
Yes, we can hear you.
Yep. Because as my background right now, I am a student, but I'm doing a master degree. As there are many species of animal and also many species of bacteria, so I'm not sure that the correlation between bacteria and animal, it will be interacted as the right. It will be their animal species will be have their selective species of bacteria or not. And the second one, is it have any factor that make the antibiotic resistance other than the gene mutations as you have present in the topic? So can you get my question? Or
I'm not fully sure I understand your question. So the second one was about antibiotic resistance genes.
Dr. Manning, I can read the question from the Q&A. So the first part of the question was, there are many animal species and many different bacteria. Do each bacteria has its own specific animal species or not?
Oh, okay. Yeah, that's a great question. In terms of the foodborne pathogens, you really have to get down to the genomic level to identify the differences. And so we know that for Shiga toxin E. coli, we see that readily in livestock, particularly cattle. We see it in deer. But we all call that a specific serotype. So we base that assessment on the serotype. And so we could say, oh, we're extracting or recovering E. coli 0157 from cows and people. That means that all of them should be able to cause disease. But we don't actually think that's true. When we look at the genomic traits, we do see that there are some, after you do some evolutionary analyses, there's some phylogenetic lineages that are more prevalent in human infections that you don't necessarily see circulating in cattle. And so I think it's a great question because that's something that we really want to understand. We want to understand which isolates are getting selected for in the environment and animals are the ones that are important for human infection, at least with regard to STEC. And so what traits might they have, genotypic traits that are important for disease in the human population? And so we still don't really know the answers to that, but I think a lot of these newer genomic tools are really helpful in allowing us to do these comparative studies.
And then the second part of the question, Dr. Manning, was, are there any factors that make antibiotics resistant to bacteria other than gene mutation? You
That's a
covered
tricky
some of those under horizontal gene transfer, I think.
Yeah, and I guess I would classify those as mutations because even though they're acquiring DNA, they're still changing the genome. And so I would say the vast majority are through some sort of mutation, whether it be horizontal acquisition of a gene or set of genes. Sometimes these plasmids have co-selection where you have multiple resistance genes on one plasmid getting transferred at once. Or, you get these point mutations. There are some bacterial species that are naturally resistant to some drugs, like lactobacillus, I think is naturally and innately resistant to vancomycin. And so I know that some bacterial species have those innate features. And it's not necessarily a point mutation that results in the resistance. Does that help?
Thank you.
Furkan, are there any more questions? If not, I have one question to Dr. Mann.
Sure.
Okay.
Ramji, go ahead, but there's one more question after.
Please, take that question, then I'll ask the question.
Stephen, do you want to go ahead, unmute and ask the question? So Stephen had a question. Do we understand the persistence of AMR genes in the absence of antibiotics from an evolutionary perspective? It seems like there would be some fitness cost to preserving it.
Yeah, that's a great question. I think we're just beginning to understand that. And so, those pathogen populations that have plasmids, large plasmids with multiple resistance genes, I think those have difficulty surviving. They're less fit because they have these plasmids. And so oftentimes you can see, sometimes they'll lose those plasmids pretty easily. There are some genes that you can get mutations, and I'm particularly thinking of those in Campylobacter. You can get mutations in the gyrase gene, which is important for DNA replication. And so if you get a mutation within that gene, then the entire bacterial cell is resistant to fluoroquinolones. And so even though you're not adding antibiotics you're not giving that patient with this new population, this new mutation, antibiotics, that population could just proliferate and multiply in the absence of antibiotics. And so just, without that selective pressure, you'll see it increasing in frequency and potentially causing disease or getting transmitted to someone else.
Thank you, Dr. Manning.
Mhm.
Ramji, do you want to go ahead?
Yeah. Thank you, Dr. Manning, for your excellent presentations. I have a quick question. You mentioned it is not only antimicrobial resistance, rather that is linked to antimicrobial use, or rather we say misuse.
Mhm.
Now, I think it is very commonly used, the terminology antimicrobial use, recently coming up to address the antimicrobial resistance. So one of them could be the education, educating the user or the doctors or whoever prescribing these, do you have any experience, which educational method is, works for, different categories of the groups they are recommending, they are using this? And I will also like to request Professor Mekong from Laos is here. We have Sangden and Wanapon and Thira Kamal from Thailand as well. Dr. Fu from Laos. After Manning's, answer, would you, one of you, jump in and share your experiences, how you are educating the public about? I would first request Mekong after Dr. Manning's, answer.
Yeah, I would love to hear their thoughts. That's not necessarily something that I've been focusing on, although I do have, 4 children, and I often go to the doctor and receive antibiotics for infections that I'm kind of questioning whether or not they might, be bacterial or viral. And so I think a lot of it is, just education in general, and that's in all industries that we see and that utilize antibiotics. I think the consumer, my oldest son never wants to take all of his antibiotics. And so really just understanding the importance and how these bacterial populations can emerge and make you sicker in the long run. I mean, it might, dampen the effect in this infection, but those genes can persist in your gut. And so the next time you get sick, maybe that drug won't work. And so I think understanding from as a user or a consumer is important, and just as important as understanding, it from a prescribing perspective, and as well as in agriculture and adding antibiotics prophylactically to prevent mastitis, for example. And so just kind of understanding the importance and potential impact that it might have, I think is important. But I would love to hear what works.
Yeah, Professor Mekong, are you in there? Would you want to share your experience, or Dr. Foog or Wanapon, any one of you or any other colleague? No?
I also
Hello, Limchi.
Yeah, go ahead.
please go ahead.
Wanapon?
Yes. One upon from now. Yes. Yes. Thank you from the expertise. Yes, talking about the antibiotic resistance in particularly for like talking about the genes resistance, something like that. Yes, I totally agree with the expertise about the resistance. Yes, in Laos, yes, when we're talking about AMR, yes, we is quite new for us as well. We start doing like the research on this, but for the deeper research, we didn't do so much. But okay, I totally agree. The gene resistant, yeah, I think because of our antibiotic use, because it's talking about the use secretly, yes, maybe misuse as well. So I think we should focus on this. And okay, in the near future, we should focus on this, and we do research on it as well. Yeah.
Yeah, definitely.
Thank
Yeah.
you.
It's-
Yeah,
yeah.
this is the information that I can share in the beginning. Maybe next I can share more about detail about this. But we need the scientific evidence. Yes. Thank you.
All right, thank you. Yeah, it is mainly origin because antibiotics are used as a growth promoter in livestock, mainly poultry and other animals, you know, so unless we restrict those use of those antibiotics in those cases, I think is going to be a huge problem. It has already been. And as the literature suggests, thank you, Dr. Manning, for your insight and sharing, you know, this thoughtful, you know, research finding.
I have two more questions from the audience. One of them is, do you consider COVID related to antibiotic resistance among foodborne pathogens in Asia?
So I guess I'm not sure of the question entirely. Do I think that the foodborne pathogens or resistance is emerging because of the link between COVID infections? Is that what I'm thinking?
I think, yeah, maybe that
Okay.
is the lead question.
Yeah, I mean, I don't know about foodborne pathogens necessarily. I do think that it's probably more hospital acquired infections that are getting affected by COVID or inaccurately treating COVID patients with antibiotics. I mean, I'm sure a lot of those cases, they do develop pneumonia, in which case pneumonia would be indicative of an antibiotic treatment. But I think a lot of those cases were presenting with viral pneumonia and it wasn't necessarily bacterial. And so I think by giving these patients antibiotics and maybe noticing that those antibiotics are not necessarily making a difference, maybe stopping treatment too early, you're really allowing that those resistance genes in specific bacteria to be selected for in the presence of those drugs. And I think that's why we might see increases in some, you know, respiratory type infections that are antibiotic resistant, just because they're now carrying these genes that are very easily transferred between individuals through coughing and sneezing. And so that kind of makes sense and would be my take on it. If I'm getting your question right,
It's just a one line question. But I also think one another way would be during COVID, a lot of the population avoided going to emergency departments and physicians and avoided going to hospital altogether and relied a lot on self-medication and more so in the developing countries where you can get antibiotics over the counter as well. So I think that could have contributed to inappropriate use of antibiotics and antibiotic resistance that you also mentioned increased during the pandemic.
yeah,
Another
that's a good
question
point.
that we have is, how do illnesses from food contamination fit into the One Health system?
That's a good question. So I guess I would say very similarly as antibiotic resistance, mainly because most of these foodborne pathogens have multiple reservoirs. And so you can find them, at least for STEC and Salmonella, you can find them in cattle, you can find them in poultry. They can be shed through feces and they can persist in the environment. In which case they can travel in water and irrigation systems, and they can survive readily there too. And so I think really understanding where they are, how they're surviving, you know, which ones are most virulent to people, at least for STEC, they're not causing disease in cattle. And it's really just people. And so really trying to understand which strains are the most important, which virulence traits are the most important for virulence and pathogenesis in people, I think will be really important. And I think that's where all these comparative studies can be really useful. You know, what do these different isolates look like from different environments? Does that answer the question?
I think it does. Thank you, Dr. Manning.
Uh-huh.
We have one last question.
Okay.
This is from Sochal Prakash Shrestha. I had conducted research project where I viewed occurrence of E. coli in captive birds of zoo in Nepal. I could not conduct the research at the molecular level to go further to genome level. What do you think the role of wildlife is as a whole in the AMR gene transfer from the wildlife?
Yeah, that's a great question. I don't think we know the extent of it and its real importance, but I do think it's probably very important. We know just from looking at transmission of these different pathogens with resistance genes across species, across livestock, across domestic animals, pets and people. I think that wildlife would be very good vectors and carriers of these genes, just like they can carry the pathogens that cause the infections. And so trying to understand, risk factors for the infections, whether it be a foodborne or food associated illness, is just as important as understanding the factors that are important for antibiotic resistance and infections. And so sometimes they're different, but a lot of times I think they might be the same. Similar risk factors are important. But
Thank
I
you,
think,
Dr. Manning.
yeah, I think that it would be really great to study a lot of these pathogens in wildlife. I know we have recovered some Campylobacter jejuni from birds that are harvesting and landing on cherry trees in northern Michigan. And, that could be important too, because they're, defecating and they're leaving the Campylobacter on the cherries and on the apple fruits that people could then potentially eat. And so I think that they play a huge role that we don't really fully understand yet.
Thank you so much for a great talk and for answering the questions.
Fuquan, there's a question from Thailand. I think shall we give a thought on this? I think
Oh,
that
yes. This one just came in. Sorry. Thank you,
yeah.
Kim.
Mhm.
What's the next step that you expect to see sentinel site surveillance, etc., at the Mekong countries regarding AMR and foodborne pathogens, etc.?
Okay. I guess I'm not probably the best one to answer that question, but I think surveillance is really important everywhere. And I think, as I mentioned, that every location, when you start picking it apart, is going to be distinct and have its own variables that might differ from other locations. And so when we just looked in Michigan, just relative to nationwide in the US, we found differences in prevalence of foodborne pathogens as well as antibiotic resistance, genes, and phenotypic susceptibility to certain drugs. And we're not sure why. It might be that Michigan has unique agricultural factors, maybe historic use of antibiotics and persistence of some of these antibiotic residues in the environment contributes to the selective pressure. But I think there are a lot of factors that might differ across geographic locations that might be really important to assess. And so I haven't had the opportunity to visit there, but it would be interesting to know, what might be unique about that region and how it might differ relative to others. I think anytime you look, you're going to find something, and it would be really important to do so.
Well, I think we're gonna wrap up today's webinar. Thank you so much. And Dr. Manning, I think the last comment you made actually is a great topic for us to talk more in next few webinars, to talk about the surveillance system and country specific characteristics, infrastructure, agriculture, environmental. So that's really a good point to start. If without any more questions, but we still encourage audience to send us questions. We'll try to address them through emails. And I want to thank you, everyone, especially Dr. Manning, to give this great talk. And it's very educational. I hope it's also inspirational to all the researchers in the Mekong region and other regions of the globe. We have seen people from various different countries out of Asia, actually. That's really great. Thanks, Jim Levy, for being with us all the time, and Wayne, Victoria, Mayfong, Stefan. Those are our, biggest champions and support, and Fook and Vanafong, and be here with us. This is really great. We hope to see you in our future webinars, and let's stay in touch. Thank
Thanks,
you
Dr.
again.
Manning.
Thank you. Thank
Thanks,
you.
Mr.
Bye
Levy.
bye.
Thank
Have
you
a
all.
great
you.
day
Bye.
and a great evening.
Thank you.
Thank you.
Great
Thanks,
evening.
everyone.
Good morning. Bye.
Yeah, our next webinar is on February 7th. Those who are still listening, it will be about surveillance.
All right. Okay. We'll send out invitations later.
Yeah.
Thank you. Bye.
Bye.
Thank you, Dean. Thank you, Dr. Fook. And thank
Thank you,
you.
Dean.
Thank you, Ting and Furkan. Thank you. We did
It's a
Thanks,
very
everyone.
good discussion. Yeah.
yeah, great discussion.